my pleasure to introduce Dr. Andisheh Abedini. She's an associate professor in the Department of Chemistry at Stony Brook University in New York. And she's also principal investigator in the New York for pancreatic organ donors with diabetes and affiliate member in the Einstein Mount Sinai Diabetes Research Center. Professor Abedini is an expert in the molecular and cellular physiology of islet amyloidosis and diabetes, and her recent work has defined the molecular properties of toxic IAPP species, which are generated during amyloid formation, and she linked their features to distinct cellular pathways that mediate pancreatic beta cell dysfunction, and that in pre-diabetes and diabetes. I just don't want to take more of your time, Dr. Abedini. It's so nice meeting you. It's so nice having you here as a speaker in our series today.
Thank you very much, Mrna, for introducing me. I also want to thank RAMS and Mrna and other meeting organizers for inviting me to share some of our work. So today I will describe some of our recent work on IAPP-induced beta cell toxicity. Our strategy has been to combine in vitro studies of amyloid formation by human IAPP and IAPP analogs with studies using both cultured cells and pancreatic islets to assess the intracellular signaling cascades that are activated in the early stages of islet amyloidosis, which contribute to beta cell dysfunction and apoptosis in prediabetes and diabetes. We complement this work with studies of mouse models and human tissue. I will begin with a brief overview of some of our work on amyloid formation, but my main focus will be on cellular mechanisms of toxicity, as this is our current focus. So the pancreatic islets are the endocrine tissue of the pancreas, where metabolic hormones are produced, as opposed to the exocrine tissue, where digestive enzymes are made. The islets are organelles that contain many specialized cell types, including beta cells, which produce and co-secrete the hormones insulin and islet amyloid polypeptide, also known as IAPP or amylin, into the circulation in response to changes in blood glucose concentration. In the homeostatic state, IAPP acts both in the periphery and in the brain to regulate satiety, adiposity, and other actions regulating metabolism and energy expenditure. However, in metabolic disease, IAPP undergoes amyloidosis by an unknown mechanism and leads directly to beta cell dysfunction and loss of islet beta cell mass and replacement of that mass with islet amyloid plaques, which is the common pathological feature of type 2 diabetes and has also recently been shown to play a role in type 1 diabetes. IAPP is a 37 residue polypeptide. It has a cis2 to cis7 intramolecular disulfide and an amidated C terminus that is required for bioactivity. This hormone is produced by all species examined to date, but not all IAPP sequences form amyloid. Interestingly, IAPP derived from species that naturally develop diabetes form amyloid. We and others, notably the Westermark and Kahn labs, have shown that islet amyloid formation is also an important complicating factor in islet cell transplantation. Islet transplantation is a promising therapeutic strategy for the treatment of both type 1 and type 2 diabetes, but the long-term success of this therapy is limited by transplant survival. We have shown that rapid amyloid formation, stained blue here, in newly transplanted human islets into diabetic mice leads to apoptosis and transplant failure. On the other hand, prevention of islet amyloid by transplantation of porcine islets, which produce porcine IAPP, which is non-amidogenic and non-toxic, significantly increases islet transplant survival and glycemic control in vivo. Identification of the toxic species in amyloidoses is a major challenge, and the nature of the toxic species is still controversial due to the difficulty of isolating pure fractions of transient kinetic species. IAPP amyloid fibrils were originally thought to be the toxic species, but recent work from our lab and others now point to prefibrillar amyloid precursors to be the culprits. It is also not known whether islet amyloidosis occurs before the onset of diabetes or is a result of diabetes. So our goals were to define the biochemical and biophysical properties of the toxic species produced during amyloid formation, identify the cellular mechanisms of IAPP toxicity, and to determine at what disease stage islet amyloidosis is initiated in vivo. Using time-dependent assays, we have shown that the cytotoxic species produced during IAPP amyloid formation in solution at physiological pH are transiently populated intermediates. In these studies, IAPP was incubated for various times and applied to rat INS1 beta cells. Cell viability was monitored by Almar blue reduction assays, which indicate metabolic dysfunction, shown here in the top panel, along with cell microscopy. The very exciting result was the observation of a wave of toxicity, where the peak in toxicity occurs before the observation of mature amyloid fibrils, as judged by TEM and thioflavin T fluorescence assays, shown here in the bottom panel. The non-toxic, non-amidogenic rat IAPP was used as a negative control. These time-resolved toxicity assays and biophysical measurements were carried out in parallel using the same peptide samples and provide proof of principle evidence that toxic IAPP species formed in solution at physiological pH are prefibrillar. The decrease in beta cell viability induced by toxic lag phase intermediates is accompanied by an increase in pro-inflammatory cytokine production and an increase in the production of reactive oxygen species and cleaved caspase-3, consistent with upregulation of oxidative stress and pro-inflammatory processes involved in IAPP-induced beta cell apoptosis. No significant upregulation of pro-inflammatory cytokines, ROS production, or caspase-3 production is observed at time 0 or at time points after amyloid formation. Our ability to monitor toxicity in a time-resolved fashion allows us to characterize the toxic species under well-defined conditions. Using a range of real-time biophysical and biological studies in collaboration with Dan Raleigh and Martin Zaney's labs, we have shown that the toxic IAPP intermediates are soluble, low-order prefibrillar oligomers with partial helical and modest beta sheet structure. And unlike the toxic species reported for other amyloidogenic proteins, they do not bind hydrophobic dyes such as ANS. Together, these studies show that while IAPP toxic species share some similar properties with the toxic species reported for other amyloidogenic proteins, they also exhibit differences. This is important because it shows that toxic species of different amyloidogenic proteins are not always similar and has important implications for rational drug design. To determine the relationship between IAPP toxicity and amyloid formation, we conducted concurrent biophysical and cellular metabolic studies using a range of conditions that alter the kinetics of amyloid formation and the length of the lag phase. These include rationally designed mutants and a range of peptide concentrations and temperatures. And what we found is that in all cases where the variants formed amyloid, there was toxicity to beta cells, but the amyloid fibrils themselves were not toxic. Variants that oligomerized but did not form amyloid were not toxic to beta cells. When we plot the length of the lag phase versus the respective duration of toxicity for all of the conditions tested, we find a striking direct correlation between the kinetics of amyloid formation and the lifetime of the toxic species. The data confirms that lag phase intermediates preceding amyloid formation are the toxic species of IAPP. The correlation between IAPP toxicity and the length of the lag phase has important implications for inhibitor design. We have previously shown that the I26P point mutation converts wild-type human IAPP into an inhibitor of IAPP amyloid formation. The thioflavin curve here demonstrates a significant effect on the kinetics. Notice that the lag phase is significantly increased in the 1-to-1 mixture. TEM studies show that I26P aggregates but does not form amyloid. The TEM images also show that addition of I26P to wild-type IAPP significantly changes the morphology of the deposits. Based on these results, we examined the effect of this inhibitor on IAPP toxicity. Our time-dependent data indicate that slowing down the rate of IAPP aggregation by addition of the inhibitor increases the duration of toxicity. This is an interesting result for two reasons. First, it further supports our findings that IAPP intermediates are toxic. And second, it confirms that an inhibitor that slows amyloid formation could actually be bad for cell viability. This is an important consideration for the design of inhibitors of amyloid formation, because drugs that lead to the buildup of toxic prefibrillar species could actually be harmful. On the other hand, inhibitors that trap toxic species could be powerful tools because they act directly on the process of aggregation and can aid in their study. There is another interesting feature on this graph, namely that I26P is not cytotoxic in isolation. This is an important additional control for our inhibitor studies, but it is also interesting because we have shown that while I26P does not form amyloid, it does aggregate. This result, in turn, shows that not all IAPP aggregates are toxic and decouples general aggregation from toxic species formation. Our understanding of the cellular mechanisms of IAPP toxicity is incomplete, but multiple mechanisms have been shown to play a role. In vivo, amyloid formation takes place in a heterogeneous environment, and multiple factors have been shown to promote islet amyloid formation. These include IAPP mutation, interactions with heparin sulfate proteoglycans and the extracellular matrices, improper prohormone processing, overproduction, covalent modification, dysfunction of clearance mechanisms, and other factors. A growing body of literature now indicates that both extracellular and intracellular IAPP oligomers contribute to islet beta cell toxicity. Histological studies of diabetic human subjects post-mortem show that islet amyloid deposition is predominantly extracellular. Transgenic mouse models that overexpress human IAPP display intracellular oligomer formation and mechanisms such as ER stress that are not displayed in cultured islet models that express physiological levels of human IAPP during islet amyloidosis. Instead, these models show that the secretion of IAPP is an important factor for islet amyloid formation and beta cell toxicity, which is consistent with an extracellular origin. And multiple downstream signaling pathways and pathological cellular events, including oxidative stress, inflammation, altered signaling, and metabolic dysfunction, have also been shown to contribute. We have focused on receptor binding because IAPP is a polypeptide hormone that binds receptors. Adaptive IAPP or amylin receptors facilitate its normal biological functions, while pathological receptors have been shown to promote its proteotoxicity. Given that the multi-ligand cell surface receptor for advanced glycation in products, or RAGE, has been shown to bind a broad class of proteotoxic ligands, including some amyloid-forming proteins, such as A-beta and prions, and that activation of RAGE upregulates many of the same pro-inflammatory processes and mediators of oxidative stress and apoptosis that have been shown to be triggered by amidogenic IAPP, we hypothesize that RAGE plays a role in IAPP-induced beta cell toxicity. Specifically, we asked whether toxic IAPP intermediates are ligands of RAGE and whether RAGE plays a role in islet amyloidosis-induced beta cell defects in vivo. We first tested if toxic IAPP intermediates bind RAGE using the soluble extracellular domain of RAGE, or sRAGE. The previous slides build a case for transiently populated intermediates to be the toxic species. If our hypothesis that RAGE could be a mechanism of IAPP toxicity is correct, then we would expect that non-toxic monomers and amyloid fibrils would not bind sRAGE, but intermediates populated at time points of toxicity would bind the receptor. And that is exactly what we observed. X-ray crystal structures of sRAGE reveal a hydrophobic patch highlighted here that contains 3 solvent-exposed tryptophans. Neither human or rat IAPP contain any tryptophans, making the intrinsic fluorescence of sRAGE a convenient probe for protein-protein interactions in this system. When we add sRAGE to human IAPP at different time points over the course of amyloid formation, we observe a wave of fluorescence quenching whose time course mirrors that of the wave of toxicity. This was exciting because it suggested that toxic IAPP intermediates and RAGE binding intermediates are the same. In contrast, we see no binding of sRAGE to the non-toxic rat IAPP at any time point. We confirmed the tryptophan fluorescence quenching results using surface plasmon resonance. SPR experiments show no evidence of RAGE binding to IAPP monomers or amyloid fibrils, but does show binding to the intermediates. These studies were accompanied by parallel TEM experiments which confirm the presence or absence of amyloid fibrils in the binding assays. We then tested the ability of the non-toxic aggregation pro I26P variant to bind sRAGE and found that, unlike human IAPP, this mutant does not bind sRAGE at any time point, producing a flat fluorescence profile similar to that produced by the one-to-one mixture of sRAGE with the non-toxic rat IAPP and that of sRAGE by itself, which are negative controls in these studies. Characterization of the end products of the reactions by difference CD show the expected predominantly random coil conformation for I26P and rat IAPP, indicating that sRAGE does not induce amyloid formation or alter the conformation of non-interacting, non-toxic IAPP variants. The data supports the hypothesis that RAGE selectively binds toxic lag phase intermediates. We carried out cell signaling experiments to determine the intracellular signaling cascades activated by toxic IAPP intermediates. Incubation of toxic intermediates on beta cells for a short period of time, indicated by the green and blue bars, provoke measurable levels of cell stress and pro-inflammatory gene expression, but not beta cell dysfunction or death. Longer incubation of the same peptide solutions on the cells, indicated by the red bars, results in significant loss in beta cell metabolic function and eventually apoptosis. Our cell signaling studies show that toxic IAPP intermediates significantly upregulate RAGE protein expression prior to detectable beta cell dysfunction, indicating that this receptor is an initiation site for IAPP toxicity. To determine the role of RAGE selectively, we used RAGE blocking antibodies and showed that they protect beta cells from toxic IAPP intermediates in a dose-dependent manner compared to anti-IgG controls. We confirmed the RAGE-dependent results in primary pancreatic islets that we isolated from wild-type and RAGE knockout mice. There are no obvious morphological differences between wild-type and RKO islets. However, genetic deletion of RAGE protects the intact islets from IAPP-induced loss of metabolic function compared to RAGE-expressing wild-type islets. We then hypothesized that sRAGE may be an inhibitor of IAPP amyloid formation since it targets toxic intermediates. To test this, we added sRAGE to human IAPP and followed the effect by thioflavin T fluorescence. The resulting time course was compared to reactions carried out in the absence of sRAGE. The results indicate that sRAGE is indeed an inhibitor of IAPP amyloid formation in vitro. Characterization of the endpoints of the reaction by TEM and difference CD confirmed the lack of fibrils and the lack of beta sheet structure in sRAGE-treated IAPP samples. If IAPP binding to RAGE is important for cytotoxicity, then sRAGE should also be an inhibitor of IAPP toxicity, and this is indeed the case. The results show that in cultured beta cells and pancreatic islets, addition of sRAGE to IAPP before or at time points of toxic species formation in the lag phase blocks IAPP-induced pro-inflammatory gene expression and metabolic dysfunction. We then conducted pharmacological studies in transgenic mice that express human IAPP and their wild-type littermates, which we treated with either sRAGE or PBS. This mouse model develops beta cell defects, but not hyperglycemia, so it is a model of prediabetes. Immunofluorescence studies in pancreas sections co-stained for insulin and amyloid demonstrate spatial overlap between islet areas with amyloid deposition and loss of beta cell area in PBS-treated transgenic mice, consistent with studies correlating islet amyloidosis with beta cell apoptosis. However, we found no correlation between the amount of islet amyloid and loss of beta cell area, consistent with toxic prefibrillar intermediates. Transgenic mice treated with sRAGE are significantly protected from islet amyloid formation and exhibit preservation of islet beta cell area compared to PBS-treated transgenic mice. In transgenic mice with islet amyloidosis, we also measured significant increase in beta cell stress and apoptosis, indicated by a significant increase in cleaved caspase-3 immunoreactivity, shown here in yellow merged islet tissue areas, and an increase in tunnel-positive beta cells, shown here in the magenta merged islet tissue areas. Transgenic mice treated with sRAGE, however, were significantly protected from beta cell stress, pro-inflammatory cytokine production, and beta cell apoptosis, and also showed improved glucose tolerance compared to PBS-treated transgenic mice. We also found significant upregulation of RAGE expression in the islets of transgenic human IAPP mice, and a significant correlation between islet beta cell RAGE area and loss of islet beta cell area, consistent with our in vitro studies with IAPP-treated beta cells and islets. Together, our in vivo studies in mice indicate that islet amyloidosis directly contributes to beta cell defects in prediabetes and is not just a marker of diabetes. We then assessed the clinical relevance of our findings in human diabetic and non-diabetic pancreas tissue from InPod, which we triple stained for insulin, RAGE, and IAPP. The results show that in non-diabetic human subjects, RAGE is expressed at low levels, but is then significantly upregulated in diabetic islets with islet amyloidosis. RAGE co-localizes with insulin, indicating expression in the beta cells, and further co-localizes with IAPP, consistent with our in vitro and in vivo findings in mice. Having established that RAGE is a pathological receptor involved in islet amyloidosis, we then followed our data to further investigate the intracellular signaling cascades activated by toxic IAPP intermediates. The binding of the intracellular domain to the formin molecule diaph-1 is required for pathological RAGE signaling. So we hypothesized that if RAGE signaling is important for IAPP toxicity, then diaph-1 must also be a key regulator, and this is indeed the case. We treated cultured beta cells with either toxic IAPP intermediates or non-toxic rat IAPP for a short period of time and found significant upregulation of diaph-1 protein expression in human IAPP-treated cells prior to the observation of metabolic dysfunction detected at later incubation periods, consistent with the idea that diaph-1 is a proximate mediator of IAPP toxicity. We then knocked out diaph-1 in mice and examined the effect on IAPP toxicity to islets. The results showed that marine islets genetically devoid of diaph-1 are significantly protected from IAPP-induced ROS production and metabolic dysfunction compared to diaph-1 expressing wild-type islets. When we compared the individual contributions of RAGE and diaph-1 to IAPP toxicity, we see that toxic IAPP intermediates have a stronger effect on diaph-1 protein expression. And strikingly, pancreatic islets isolated from diaph-1 knockout mice are fully protected from IAPP-induced ROS production and metabolic dysfunction, while RAGE knockout reduces toxicity compared to wild-type islets. Together, the data indicate that diaph-1 mediated signaling is critical for IAPP-induced oxidative stress, which in turn is a proximate mechanism linked to the development of beta cell and islet inflammation, metabolic dysfunction, and apoptosis, and that diaph-1 is an essential regulator of multiple mechanisms of IAPP-induced islet beta cell defects. To summarize, our time-resolved biophysical and biological studies provide proof of principle evidence that prefibrillar lag phase intermediates, and not the amyloid fibrils, are the source of IAPP toxicity. Our studies with non-amyloidogenic, non-toxic IAPP variants show that not all IAPP oligomers are toxic. Toxicity is conformation dependent and not simply a result of forming an aggregate. We showed that inhibitors of amyloid formation can, in some cases, trap lag phase intermediates and prolong toxicity. We demonstrated that islet amyloidosis, extensive beta cell stress, islet inflammation, and beta cell dysfunction occur in prediabetes before the onset of hyperglycemia and diabetic complications, consistent with the idea that islet amyloidosis contributes to the progression of diabetes and is not just a marker of the disease. Our studies show that RAGE activation by toxic IAPP intermediates is one of multiple diaph-1 mediated mechanisms of IAPP-induced beta cell toxicity. And our data also indicate that diaph-1 is an essential regulator of IAPP-induced islet beta cell oxidative stress and metabolic dysfunction. Together, these findings define the toxic species of islet amyloidosis and demonstrate that there are multiple mechanisms of IAPP-induced islet beta cell toxicity. Specifically, our work is the first to demonstrate the key role of the RAGE diaph-1 signaling axis in IAPP-induced beta cell and islet defects in prediabetes and type 2 diabetes. I would like to thank my colleagues and collaborators, particularly the students and fellows who worked on the projects in my group, Dan Raleigh and Martin Zaney's labs, who collaborated with us on the biophysical studies, Bruce Verscher and Annette Plessner at UBC, who collaborated on the islet studies, Anne-Marie Schmidt and Jackie Lornier, who worked with us on the RAGE studies. Thanks to our funding support and to the NPOD organ donors and their families who make our translational studies possible. And thank you all for your attention.
Hi, thank you so much for the exciting talk. That was really great. And you have so many questions already. Please, just to the audience, post your questions in the Q&A portion. I will read them from the chat, but just to remind you to post the questions in the Q&As. So the first question is from Rams, who says, great talk. Andy, how do the aggregation kinetics, particularly the lag time of human IAPP, compare for in vitro compare and cellular conditions. So how I'm guessing that's how they're compared both in vitro and cellular conditions, the aggregation kinetics.
So we have looked at IAPP amyloid formation kinetics, both in neat buffer and also in RPMI in cell culture. And we find that in the buffer condition, it really depends on the ionic strength of the buffer. For simple salts, complicated effects are observed. Dan Raleigh's group showed that IAPP amyloid formation is sensitive to the choice of anions, even at moderate salt concentrations, like 25 millimolar. And anions that interact more strongly with positively charged ions lead to faster amyloid formation. The origins of the effects are not yet fully understood. So that said, the aggregation kinetics in neat buffer depends on the type of buffer that is used. In the situation of cell culture media in RPMI, it's very interesting because there are two competing effects. What we found is that, first of all, the increase in temperature, 37 degrees, is going to increase the rate of amyloid formation compared to experiments done at 25 degrees, which is usually the temperature that we use when we're looking at aggregation kinetics in neat buffer. However, RPMI, if it's fully supplemented, also contains BSA, and BSA is an inhibitor of amyloid formation, we found. We haven't published this, and I believe Dan Raleigh's lab is looking more closely at the interactions and the effects, but so in a way it balances itself out. And so in our hands, what we find is that there really isn't too much difference between doing amyloid formation kinetics in tris buffer versus doing the amyloid formation kinetics in the RPMI in cell culture media.
Thank you. So I'm going to continue with the Q&A session. A question from Anu. Does the cytosolic domain of RAGE bind to intracellular IAPP monomers or oligomers?
Does the intracellular domain of RAGE bind
Does
to...
the cytosolic domain of RAGE bind to intracellular IAPP monomers or oligomers?
So the intracellular domain of RAGE binds to formin molecule diaf1, which is required for intracellular RAGE signaling. Now, there is an increasing body of literature that now points to both extracellular and intracellular oligomers to contribute to islet beta cell toxicity. But whether the intracellular oligomers bind to RAGE, I don't believe anyone's looked at that, but that's a very interesting question.
Thank you. The next question is from Ifat. Excellent talk, Andy. Can you distinguish between the polymorphic oligomers to really indicate which of them are toxic and which of them are not? Have you thought of doing that?
So it's difficult to isolate pure fractions of transient kinetic species, and IAPP toxic intermediates are almost certainly a heterogeneous mixture of soluble, highly dynamic, flexible oligomers. And we have done photoactivatable cross-linking studies, and we find a distribution of oligomer sizes predominantly dimers and trimers, but we detected up to hexamers. But when it comes to the polymorphs structurally of the oligomers, no, we have not looked at that in that great a detail to differentiate between polymorphs of oligomers. That's a good question.
Yeah, thank you so much. Then I'm switching to the chat area. We got lots of questions in the chat. The one is from Dr. Rohit Kulkarni. A very interesting talk, and there are actually two questions. The first one are there polymorphisms in IAPP-1 and RAGE that correlate with type 2 diabetes in humans? And the second question is, can one dissolve toxic fibrils by beta cell specific delivery of sRAGE or related molecules? In other words, what are the therapeutic options for type 2 diabetes?
So in terms of the therapeutics of sRAGE, free sRAGE binds directly to RAGE ligands and competes with membrane-bound RAGE for ligand binding. It essentially acts as a dominant negative inhibitor. The first question was it what type of isoforms of diaph-1 are involved in the human disease?
Yeah, are there polymorphism of the IAPH, yeah, that correlate, and RAGE that correlate with the human disease?
So there are different isoforms of this protein. And diaph-1 has been implicated in various pathologies, including seizures, blindness, deafness, and now our work, metabolic disease. And Anne-Marie Schmidt's lab has also shown that diaph-1 plays a role in metabolic diseases as well. There's a large body of literature on the role of formin molecules and cellular signaling, and diaph-1 interacts with numerous proteins in the regulation of multiple cellular signaling pathways, including RAGE, that are involved in nucleating new actin filaments and the regulation of oxidative stress, cytokine and chemokine production, alterations in cellular migration, proliferation, and survival in several different cell types. But nothing is known about the functional role of diaph-1 signaling in beta cells or islets, particularly in the setting of diabetes. And we are now investigating this pathway in more detail in both healthy beta cells and in the setting of islet amyloidosis and metabolic disease.
Yeah, thank you. So I have two questions which are coming, well, they are on the realm of the aggregation of IAPP, and it's again coming from the chat box. It is from Martin Moshu, who says that one aspect of the oligomer formation that is intriguing in many systems is they are surprisingly short-lived, particularly when compared to the slow chronic progression of amyloid diseases. Do you have any insights? So do you have an idea of how long-lived are these oligomers? And yeah, just pass it to you.
So certainly, diabetes is a progressive disease, and the in vivo amyloid formation takes place in a heterogeneous environment. So our studies indicate that islet amyloidosis induced beta cell defects are initiated in the pre-diabetes stage, before the development of full-blown hyperglycemia and diabetic complications, and before the accumulation of significant islet amyloid deposits. So given that pre-diabetes occurs much earlier and diabetes is a progressive disease, and it's actually now thought that the driver of diabetes is not beta cell death, but actually beta cell dysfunction. And so depending on clearance mechanisms and various other factors that could be contributing to the rate of aggregation, and that would probably differ, in different disease stages. So it's probably definitely in diabetes, it's going to be a much longer rate of amyloid formation kinetics than what we observe in the test tube. So yes, it's much more progressive. It's a much slower process in vivo than what we would observe in vitro studies.
Yeah, thank you. So I'm kind of picking up the questions randomly to stay on the topic. The other question, which is coming from on Gary, is the best to inhibit IAPP toxicity is this to preserve the monomeric species. So that's down the therapeutics of IAPP.
In terms of the
do you think it's the best to, the question is whether it's the best to work on preserving monomeric species in the monomer state?
So we are convinced that targeting the toxic soluble pre-amyloid intermediates, which cause beta cell dysfunction before the formation of amyloid fibrils, and which appear to also be the building blocks for amyloid fibril formation, are critical targets for therapeutic intervention in islet amyloidosis. Since we have shown, and Peter Butler also has noted that islet amyloidosis appears to be occurring in prediabetes, and people who have diabetes, who are diagnosed with diabetes, actually have diabetes for a much longer duration than when they're diagnosed. So they have it for a longer period of time before they're finally diagnosed to have it. So I think it would be hard to try to prevent toxic species formation. But if we can prevent their formation, if we can identify people before they can become pre-diabetic, then that would be the ideal case. However, since people find out that they're pre-diabetic, or they find out that they're already full-blown diabetic, then chances are that those toxic species are present in the body. And so perhaps a combination of preventive strategies, as well as strategies to target the toxic species that are already there, whether to sequester them and prevent them from interacting with cells, or bind them and lead them to a different protein folding pathway where they're no longer toxic, could probably be beneficial.
Yeah, thank you. So I'm going to ask one question from Christian, and for the sake of time, we will go over the remaining of the questions after the second talk. And hi, Andy, is RAGE upregulated only in beta cells of type 2 diabetes patients, or is RAGE also upregulated in other cells?
RAGE is expressed ubiquitously, so it's expressed in all cells that have been examined to date, and it does become upregulated in inflammatory states. And so it's not just in the state of diabetes. It's been implicated in Alzheimer's disease and cardiomyocyte dysfunction and various other inflammatory diseases and multiple cell types.
Yeah, thank you
for
so
that
much.
question,
Yeah,
it's a good question.
Yeah, that's a cool question. So we will continue with the remaining questions. after Ralph's talk. So thank you so much. And if you don't mind to stay longer so you can answer the remaining questions. All right. Hi, Ralf. Do you mind uploading your slides? Great. Everything looks perfect. Good day, everybody. This is the second talk in our ZOOMinar session. Will be by Dr. Ralf Langen, who is the professor of physiology and neuroscience in University of Southern California, Los Angeles. He's also a director in Protein Structure Center, Zika Neurogenetic Institute, and director of the PhD program in the medical physiology, co-director of the PhD program in medical biophysics. His main research interests are in the basic areas of protein folding and misfolding, with particular emphasis of the involvement of membranes in protein folding and aggregation. Very nice having you today, and it's your turn. Nice seeing you again.
All right. Thanks, Magda, for the introduction, and thanks to Rams and all of the organizers for allowing me to present my research here. So over the last several years, we have been interested in the mechanism by which the Huntingtin protein misfolds. But what I want to talk about today is our work on trying to find conformationally specific binders. So the Huntington's disease is caused by polyQ expansions in the Huntingtin protein. The full-length Huntingtin protein is actually a huge protein. It contains over 3,000 amino acids, but the polyQ region that is being expanded is actually in the first exon of the Huntingtin protein. So N-terminal fragments, like the first exon, actually accumulate in disease, and they accumulate in aggregates. So they're generated by proteolysis and aberrant splicing. So much of our work has been done on the Huntingtin exon 1 protein, which contains 3 domains. It contains the N17, which is the N-terminal region, 17 amino acids in this N-terminal region. It contains the polyglutamine region. And then at the C-terminus, it contains a region that is very rich in prolines, and we refer to it as the proline-rich domain. Over the years, a lot has been learned about the aggregation pathway of exon 1 from monomers to fibrils. And one of the early steps, if one just looks at the Huntingtin exon 1 protein aggregating in solution by itself, is that it forms a helical intermediate. And this helical intermediate has already been proposed by early work from Ron Wetzel. It had been, additional evidence has been generated by Judith Friedman's work. And in 2018, we published a study where we looked at the aggregation of the exon 1 protein, and we found that early on, using CD, we can find helical structure forming before the beta sheet structure forms. And we looked at where these helical structure formed using EPR spectroscopy. We found that it is in the N-terminal region, N17. And Marius Kloor's group has recently published a study that actually suggests potential arrangements of these helices in these oligomers. Then upon aggregation, what we see is that beta sheet structures form. So there are a series of aggregation steps that lead to the formation of protofibrils, and then there are different types of fibrils. So a lot of the work, a lot of what we know about the fibril structure comes from work by Patrick van der Wel and also from cryo-electron microscopy. Patrick's group found that the polyglutamine core forms these cross-beta structures, and he also found that the N17 region actually is still retained as a helical structure. among many other things that he found. We have looked at fibrils also using structural biology tools like EPR and in collaboration with Ansgar Ziemer's lab using solid state NMR. And we found, one of our findings was that the proline-rich domain is facing outward like bristles. It actually is a highly mobile region that contains a lot of polyproline 2 structure in addition to some random code structure. And these proline-rich domains shown here in green are actually sticking out into the solution. And what happens is that these fibrils are unbundled for some period of time. And over time, these proline-rich domains start to interdigitate with each other. And it causes a serious bundling of the proteins. And as we heard in a previous talk, not all aggregates are toxic. And we found that these highly bundled fibrils are actually non-toxic, whereas the unbundled fibrils have toxicity. And if you look at the most unbundled fibrils and really short unbundled fibrils, which we refer to as protofibrils in this publication, they have the highest degree of toxicity of these fibril types. So in addition to this aggregation pathway, like many other amyloids, one can also add seeds and circumvent this pathway and significantly accelerate aggregation. Then there's a third pathway, which is also found in many other amyloid proteins, and that is the ability of membranes to catalyze misfolding. We were able to show that certain membranes can significantly accelerate misfolding, and that seems to be mechanistically done by the N-terminal 17 amino acids, which dock the protein onto the membrane surface like an anchor, where then aggregation can occur. And this can significantly accelerate the misfolding process. And this is also something that where Justin Leiter's lab has made many contributions. So there's a fourth mechanism of aggregation. And that is one pointed out by Helen Seibel's lab, and this was published a few years ago in Molecular Cell. She found that, or her lab found that the Huntingtin protein can undergo liquid-liquid phase separation, and which ultimately causes the formation of fibrils from a transition from liquid to solid. So what we would like to do is trying to see how all these mechanisms that we can come up in the test tube, how they come up, how we can find them in cells and in animals. So what we think would be very helpful is to have some conformationally specific binders. If we had them, they could allow us to address questions like, do these structures exist in vivo? When and where do they exist? Which pathways are relevant in vivo? Binders might also be useful as diagnostics in patients. Then we might be able to prevent the formation of toxic species, or even if we can't prevent their formation, we might be able to wrap up these toxic species and protect from toxicity. One way would be to use antibodies, but we feel that current antibodies do not have much resolution. Mostly they allow us to distinguish between monomeric and aggregated species. So in order to address this, we have been, over the past few years, been looking into 3 different approaches. We have been working on antibodies. So this was done is currently been going on in collaboration with Ali Kashnan's lab, where we're trying to look at existing antibodies and try to see whether there are some that are highly conformationally specific. We've also been able to make defined conformers, and we're currently trying to identify antibodies, hopefully being able to get conformationally specific antibodies. We have looked into small molecules, and what I want to focus on for the rest of the talk is our work done on mRNA display, where we have been able to generate peptides together with Rich Roberts and Jeannie Chen, my colleagues here at USC. So one of the nice things about the mRNA approach is that if we can make it and link it to a bead, we can try to get a binder. So we generate the species in our lab, and then we can work with the Roberts lab, the lab that pioneered the methodology, to find peptides that bind to the target. So what you see here is exon 1 bound to beads. And in red, you see a peptide that binds to the beads. So we can now pull down the peptide with the beads and then try to find out what peptides was this. So the key here to this technology is that the peptides are linked through puromycin to an RNA tag, and through some molecular biology, it's possible to figure out through the analysis of this RNA tag which peptide came down. And then it's possible to generate a library of all the peptides that came down with the beads, to in vitro translate those, and then to go through multiple rounds of binding and then get stronger and stronger binding. Ultimately, we can sequence several millions of clones and try to find the most promising sequences. Then after we have peptides, we're interested in trying to find out the binding specificity, their effect on misfolding, as well as the structure of these binders. And of course, we want to know whether they could be potential biomarkers or whether they could be misfolding inhibitors. Okay, why, you might ask, why do we want to use mRNA display? Well, there are really three key advantages of this methodology. One is that we can produce, in the lab, we can produce very well-defined metastable biochemical targets. And we can load them onto beads. We can then use EPR spectroscopy to actually show that the structure of these conformers that we have on beads is still the same that it was in solution. And that type of control is something we don't have when we make a conformer and inject it into an animal. We can later see what we got, but it's much nicer if you have, especially if you have a metastable species, to be able to control it and to have it on a bead that you can add in a test tube. Then another advantage of mRNA display, for example, over phage display or other methods, is that we have a very large number of independent sequences. So we can test about 10 to the 12 independent sequences. And by having this very large number of sequences, one tends to get higher affinities because one has a much larger pool of sequences to choose from. And then also we end up with peptides that tend to be stable in the cytosol, which is something that is not always a given with an antibody. Okay, so the first target that we chose were these protofibrilla, these earliest fibrillar species, and we're interested really in all of the species, but we chose it because it's an early misfolding intermediate, it's highly seeding competent, and it might be common to different pathways, perhaps. So here's the experimental results. So when Kaori Noridomi, who did this work, used the protofibril containing beads, she went through several cycles of finding these peptides, and with each cycle, the number of peptides that bound to the beads increased. We then sequenced those, and we found several families of peptides, which we call HD peptides. And we have extensive sequence information for each of these families. So we have several million sequences. And we found that two families, which we refer to as HD1 and HD8, are the best binders, and they also have the highest affinity, and I'll mostly refer to work that we have done on those binders. So the first question is, can these peptides be biomarkers? So one of the experiments Kaori did early on is to test the specificity. So she used beads that either contain nothing, just the beads alone, that contain the protofibrils, that contain monomer, or that contain alpha-synuclein fibrils. And she only saw binding in the case of protofibrils. There was no binding to alpha-synuclein. There was also no binding to A-beta. And this is important in a sense because some of the other molecules that are currently used potentially as PET ligands or as biomarkers have somewhat of a cross-reactivity with other amyloids. So we have done a lot more EPR work on this, which I won't have time to show you, but I'll just summarize this. So we found that HD8 and HD1 family members of the peptide preferentially bind to protofibrils. They do bind to unbundled fibrils as well, but they seem to have relatively a reduced stoichiometry, and it's very difficult to detect binding to bundled fibrils. Then we've done some fluorescence correlation spectroscopy work, and we've been looking specifically for what are the smallest species that these peptides can bind to. And we were quite surprised to see that they can actually, or not surprised, we were happy to see that they can bind to 10 to 30 mers of exon 1 already. So they can bind very early on in the aggregation pathway. Then we wanted to know where on exon 1 is the binding. And for that, we used N-terminal as well as C-terminal trackations. And we found that we can take off the N-terminus and the C-terminus. And the binding occurred, we could conclude that the binding is actually in the polyglutamine region. So then we looked at the binding affinities. It is common to get nanomolar affinities from the mRNA display methods, and indeed, some of the monomer peptides had affinities down to about 300 nanomolar. One thing we found is that when we linked peptides together, the affinity went down to about 50 to 100 nanomolar, and it was remarkably even possible to link HD8 and HD1, so two peptides with very different sequences together with different linkers, and that even enhanced the binding, which suggests that these peptides have very adjacent binding pockets. We could go to trimers and tetramers, and we found even lower KDs. They are in the low nanomolar range. So these are very useful, super high affinity binders. But we have done most of the work with the dimers because they tend to be, they have no issues with solubility, whereas in particular, once you go to a tetramer, solubility could become an issue. So the conclusion from this was that the beta sheet polyQ surface is a highly repetitive surface with many adjacent binding sites. So in order for this aggregation, or in order for this binding to be much stronger upon multimerization, there must be adjacent binding sites. And it's probably somewhat intuitive why this would be the case. Any amyloid fibril is a highly repetitive surface, but the polyQ core is even more repetitive because you have the entire surface is made out of glutamines. So next, we wanted to know whether these peptides recognize misfolded exon 1 in cells. For that, we used hex cells, which were transfected with exon 1 with 72 glutamines. And even in the phase contrast, after about 24 hours, you can actually see these aggregates. When the HD8, HD1 dimer peptide with fluorescein was added, you can see these green dots. So the peptide actually does co-localize with the aggregates. In addition, when we zoom in and go to higher laser power, what you actually see is that in addition to the aggregates, you can also see smaller species, which actually turn out to be, for the most part, perinuclear in nature. There is reasonable overlap with the Php1 antibody. The Php1 antibody is an antibody that binds to the proline-rich domain. It actually binds kind of to the outside of the proline-rich domain. And we see similar species being detected. There are actually some differences in binding, and if anybody's interested, I can comment on this a little bit more. But the main point of this slide here is that there are binding sites that were obtained on recombinant protofibrils that these peptides bound to, and they exist in cells. So then next we wanted to know whether these binding sites also exist in transgenic mice. So we work with the R61 mouse, and this is a mouse where exon 1 is overexpressed. We looked in control retina and control cortex, and we saw no deposition that could be detected with the peptide. Whereas here in green, in the transgenic animal, you see in the retina as well as in the cortex, you can see aggregates that can be detected. These aggregates seem to be cytoplasmic, as well as here where you see these asterisks, these aggregates seem to be in the nucleus. So this means that the HD peptides can recognize exon 1 aggregates in these R61 mice. So even in vivo, we can use our binders and we can detect the structures. So sometimes in the field, the question is how relevant are these mice that express exon 1, whereas in the actual disease, you have the full-length protein. And the full-length protein then gets processed into smaller N-terminal fragments. So we wanted to know whether our work was, whether the peptides also recognize aggregates in a mouse that expresses full-length protein. And what we were able to show is that after 48 weeks in this ZQ175 mouse, we can see puncta, defined puncta, whereas even when you crank up the signal, there's very little to be detected in the control animals. So we find that these poly-Q beta sheet aggregates are present in full-length Huntingtin mouse as well. So then we wanted to know at what time can we start to see the aggregates. And it has been reported that the early aggregates are being seen after about 4 to 5 months. And we looked at animals even earlier, after 12 weeks, and we were able to see some aggregates already after 12 weeks. So detection is earlier than what has been described with antibody staining, and we confirmed this. We were not unable to detect these aggregates with a number of currently used antibodies. So we're currently testing different regions of the brain and other model systems to see whether this is a common thing that we might be able to detect earlier aggregates in a number of systems. The next question I want to go to is whether the HD peptides are aggregation inhibitors as well. And so for this purpose, we looked at each of the 4 aggregation pathways that I showed you earlier. So first, I want to just briefly go into what happens in the unseeded and in the seeded aggregation pathway. And to do this, we've looked at EPR-based readouts. This is based on the introduction of a spin label at position 35 in the polyglutamine region. And earlier studies that we had published showed that the aggregation of the beta sheet formation of the polyglutamine region coincides with the immobilization of residues in this region. So we can use this EPR of the spin-labeled protein to determine aggregation kinetics. So here we just screened a number of different peptides. And what's shown here is the EPR amplitude for different conditions. So first we had a condition where we added no peptide, then we added an HD8, HD1 dimer. A and B indicates different linkers. And then we made an HD1, HD8 dimer, and then we looked at HD1, the monomer peptide alone. So at time 0, all amplitudes were at 100%, meaning that no aggregation had occurred. After 1 day, in the absence of peptide, aggregation was nearly complete, whereas in the presence of all of these dimeric peptides, the aggregation had just begun and wasn't anywhere near complete. The monomeric peptide inhibited as well, but it was not as potent as the dimeric peptides. So then after 4 days, we still were able to see that for the best of the dimeric peptides, more than 80% of exon 1 was actually still not aggregated after 4 days, whereas the control, as well as just adding HD1 monomer peptide, had aggregated completely. So we've learned that different linker geometry and arrangement of dimers actually matters, and we have a lot more additional data on this. And I won't be able to show you the data for interest of time. Oh, also another point is the dimers are better than the monomers. And I won't be able to show you the data for seeded aggregation, but they're equally potent inhibitors of seeded aggregation. So 2 of the pathways I mentioned can be inhibited already by these peptides. Next, we wanted to test whether the peptides can inhibit against liquid-liquid phase separation mediated aggregation. So to do this, we first tried to repeat the experiments done by Helen Seibel's group. And we were able to make exon 1 with an RFP label on it, and we're able to show that in 10% pig, having 10% pig in a buffer, we were able to generate these liquid phase separated forms, which fairly rapidly transition into fibrils that are shown here in through electron microscopy. We can then time resolve this, again, using a spin-labeled Huntingtin exon 1. And what we find is that in the absence of peptide, aggregation is very rapid, whereas if we add peptide, the aggregation is strongly reduced. So also in this particular aggregation pathway, the peptides can inhibit. So how about membrane-mediated aggregation? So for this, I will show you some of our thioflavin T data. And in the absence of peptide, aggregation occurs very quickly. Almost in half an hour, it's complete. And even though the effect is not as pronounced as in the case of the other three mechanisms, also here do we find that the peptides inhibit membrane-mediated aggregation. So they inhibit, our binders inhibit all four different aggregations pathways, and that means that at least some of the intermediates on these pathways must be structurally related. We're actually screening more and more peptides, and we're trying to see whether we can find peptides that can selectively inhibit one pathway over the other, and thus will be a nice tool then to look at aggregation in a biological setting. So we then looked at aggregation inhibition in cells using these peptides. And what we did here is we used hex cells, which were co-transfected. The hex cells were transfected with exon 1 Q72 RFP, so they will appear here as red. And they were either co-transfected with GFP alone, or they were co-transfected with GFP HD8, HD1. And what you can see, and it's shown here in the black and white image, that these red puncta are forming in both cases, but the puncta are much more prominent in the absence of the peptide. And this is quantified over here, where you can see that when just GFP is co-transfected, you see lots and lots of puncta. And when there's co-transfection with the GFP dimer peptide, the puncta are reduced. So it strongly inhibits puncta formation and aggregation in cells. And I won't be able to show you all the data, but we also found that there's protection from toxicity in striatal cells. So how could we take this toward inhibition in vivo? Well, we are not there yet, but we have just started to make a viral vector, which we've been applying using systemic administration by retroorbital injection. So far, we know that it's possible to get expression in more than 60% of the brain, and we have not been able to find any toxicity so far of the peptide. Then there are other ways, which is to possibly modify the peptides or generate small molecules. And to do this, it would be nice to know which residues are important, what is the structure. And we've actually obtained extensive sequence information for each of the families of peptide binders, so we know which residues are conserved. And we've been able to confirm that these residues are important using alanine scanning experiment as shown here. For example, this is in HD1, where we know residue 2, 3, 6, and 9 are important, likely in what seems to be a helical structure. We're now confirming the structure using EPR spin labeling and trying to find a secondary structure as well as which residues are buried. And another point I would like to make is that we can also use this, these peptides as a platform to search for small molecules that bind to the same sites that the peptides bind to that are inhibiting, but then displacing the peptide from the fibrils or from the protofibrils. So to summarize this, we've been able to make high affinity conformationally specific peptides from mRNA display. We're currently extending this to all the other conformers. It detects Huntingtin in cells and in animal tissue. So far what I've shown you are really antibody-like staining experiments. But what we're also working on, what is likely going to work out as well, are ELISA experiments where we can take serum or CSF and then detect these toxic aggregates. And for this in particular, it will be very useful to take the binders that have really low nanomolar affinity to be able to pull down aggregates at very low concentration. The peptides are potent inhibitors of misfolding, and the parallel pathways suggest that they are structurally related intermediates, the inhibition of the parallel pathway. So I've already pointed out on the slides the people who did the work. So I just want to acknowledge funding from CHDI and from NIH. And the work was done in collaboration with the Rich Roberts lab at USC and Jeannie Chen lab at USC, and she did the cell work. And that's pretty much it. Thanks for your attention.
Thank you so much for your great talk. It's very exciting to see that you have such a strong inhibitors of fiber formation of polyQ. Can you elaborate a little bit more on how your peptides compare to the antibodies, and whether you have tried to see if they can cross the blood-brain barrier? If they're crossing there, like do you
Right. So the peptides are very different from the antibodies. So it's actually so I guess there are two questions to this. So peptides bind to the polyQ, and all of our efforts to make conformationally specific binders for the fibrils have so far yielded binders to the proline-rich domain. And the proline-rich domain is something that, for some reason, we think it doesn't actually even change the structure upon going from a monomer to the fibril. So what these antibodies seem to be doing is that they hang on to two bristles at a time and probably have higher affinity for fibrils because you have this sea of bristles rather than detecting them in a monomer. And we've actually tested this by putting the proline-rich domain onto proteins like synuclein, made a fusion protein, and we can actually now trick the antibodies to recognize aggregates that by just presenting bristles of proline-rich domains. So that actually seems to be one of the mechanisms. They are still working. They still work. They work to detect aggregates. The other thing is antibodies have a problem with aggregates in general because they're too big and they can't really get to the polyQ quite easily. So the peptides can go to the polyQ. They can go through this deep, forest of bristles essentially and go all the way to the polyQ, and they can sense the actual conformation that occurs. With respect to delivery, we have thus far not tried to get these peptides past the blood-brain barrier. So we have so far used delivery of peptides through viral vectors, but it would have to require some modifications of the peptides to get them past the blood-brain barrier toward the target cells. It might be possible to get small molecules to mimic this. So this is another avenue that we're going after right now in parallel. But the primary step that we're taking right now is to express it using viral vectors.
Yeah, thank you. So there's two quite similar questions, and I'll just ask them also in a sequence. The first one is from Vijay, who says, "Hi, Ralf, good to see you, wonderful talk. Are these peptides aggregation prone, or do they phase separate by themselves? If the peptides inhibit LLPS mediated aggregation, I would guess they partition into the droplets. Is this the case?"
Yeah, that's correct. So based on our analysis so far, we know that the peptides mostly are monomeric in solution. We may be able to drive them into a phase-separated state. That's possible. So we still have to look into this. This is a very good question. And so far, we have mostly looked into trying to understand or trying to see whether they can inhibit all these different aggregation pathways. And that has been our main goal at the moment. But so the answer is we don't quite know yet the precise mechanism. So when we look at co-localization of the peptides with Huntingtin exon 1 phase separating, we see that the peptides go into, they co-mix with exon 1 in those droplets. So yes, they go in. But what we're trying to find out is whether they're being recruited by beta sheet aggregates that are forming in those droplets, or whether they're already co-mixed. And these are experiments that are ongoing right now. So I don't want to comment on that. This is about as far as I want to go on this question right now.
Yeah, thank you. So the other question is coming from David Eisenberg, who says, Ralf, how many residues in your peptides? And the second part of the question, do they also aggregate? So how many residues are in the on the average in your peptides? And also I would add, how big is the linkers? Where you've been talking about testing different linkers, how big are approximately the linkers?
Right. So the actual peptides are 10 amino acid peptides. And then we have played with various linkers of 5 amino acids, 10 amino acids. Sometimes we put something in front, sometimes we put something in the end. So these peptides alone, yes, but the building block is really a 10 amino acid peptide that is screened for. The question whether they aggregate, so far they have been reasonably well behaved and staying in solution monomeric for extended period of time. So we can put spin labels on them and we can actually follow their dynamics. And we've actually used these spin labels to determine whether they bind to fibrils or whether which species they're binding to. So we can actually detect whether there's a change in the structure and a change in the motion of the peptide. So when you go to dimers, they're still reasonably well behaved. However, when you go to the tetramer portion, then it's still possible to put it on beads and to use it as a capture reagent, but it's a little bit more complicated now to handle that peptide in solution. So that could possibly aggregate, but it still would be a good capture reagent for something like an ELISA purpose.
Yeah, thank you. And do you know what is the half-life of the peptides have you tested them in an animal models?
We have not systematic. That is something that is definitely of interest to us, and we clearly want to do that. So we don't know that at the moment. We do know that we see that these peptides are, at least in a cell model, that they are effective and that they prevent aggregation. So they must be at least available for long enough to be able to prevent aggregation. We have looked at the stability in serum, and that stability we could barely detect any degradation in serum. So these studies are ongoing. But we also have been using it as a fusion protein with GFP, and that might have actually helped its stability as well.
Yeah, thank you. Yeah, have you thought of using the amino acids to see whether they would have the same, because this can help with the stability of the peptide.
Mm-hmm. Yeah, I mean there's several ways. I mean, we could use that. There are also ways to actually use proteolytic digestion during the screening process. So during the actual mRNA display method, one can add proteases and actually select for the ones that are resistant to proteolysis. But there are many ways of doing this, and this is definitely one of the things we're interested in.
So and I assume you isolated a lot of peptides, and again, asking down the question of the behavior of these peptides, which are they all behaving well, or you are in general, meaning not much aggregation?
Yeah, so the total number of peptides sequences looked at was well over 1 million. So there were a large number of peptides. And then what our collaborators do is to look at the overall hydrophobicity and predict which ones would be soluble. And so more than half of the hits are being just eliminated right off the bat because they just seem to be problematic. And so we then just work with the families of peptides that we expect to be soluble.
Yeah, thank you. Thank you so much. So I guess we can, we have a question just came in from Aphrodite. What are the key sequence requirements for inhibitory effect of the peptides?
Well, that's a good question. So we find that peptides with completely unrelated sequences can be very potent inhibitors. But what I think the key is, it's more about the binding pocket. I think that inhibition seems to occur when a particular binding pocket is taken up. So it really is the location of where the binding occurs, and that can be accomplished by a number of different sequences. So each peptide has their own amino acids that seem to be important. So, and the reason why we are convinced that it's the pockets, because we can test other known inhibitors and see whether whether our peptides bind for the same type of pockets that other inhibitors bind to. And so far, everything that we have tested seems to be binding to the same sites. So it seems to be mostly about the sites. And one of the things now that we would like to know, is it the site plus some blockage sterically somewhere, or is it just binding to the site that is enough in order to prevent aggregation? So this is something that we are now playing with, modifying the molecule, putting like a tag on one end, on the other, and seeing whether we need to size it to a certain size.
Yeah, thank you. And you have a question from Patrick. Very cool results. Do you think that the inhibitory activity might relate to the blocking of secondary nucleation as you saturate the polyQ fiber surface?
I think that's so these studies are ongoing. And so right now we're doing very systematic studies where we do concentration dependence, both for the protein as well as the inhibitor. And we're trying to use the Knowles type math models and using a MiloFit to try to get at this question. Intuitively, I think it makes sense that secondary nucleation could be important here, because we actually find a very high binding stoichiometry. So we can find that almost one peptide can bind to for every 2 or 3 exon 1 molecules. So there's a very high binding stoichiometry. So this really could indicate that a good fraction of the fibril surface is being covered by the peptide. Of course, that doesn't mean that every peptide is inhibitory. Maybe you need only 1 out of 50 peptides to be inhibiting and the other ones bind. But it's quite a good likelihood that binding to the surface is very important. So yeah, it's a very good question.
Yeah, thank you so much. I guess now we can switch to the questions to both speakers. And for those of you who want to join the panel, please raise your hands so we can let you in. And we can, Andy, I was seeing that you have been answering the questions from the Q&A portion, which I posted on the Q&A. But have you been keeping a tab on the ones which are posted on the chat? I can read them for you. Hi, Andy.
Sorry, I was just finishing up typing an answer to one of
Yeah.
the questions.
So did you end up I saw that you answered the Q&A, but I don't know if you answered the questions from the chat. We can go through these questions.
Yes, I answered all of the questions in the Q&A,
Uh-huh.
but not yet all of the ones in the chat.
Yeah, so have you started or not? So I have the chat for me. I can read them for you, or you would just rather to?
No, it would be great if you could read them. Thank you.
Yeah, so the question was, there's this one question from VJ who says, Andy, wonderful talk. I'm curious about the state of disulfide bonds present in IAPP during aggregation and the interaction with RAGE in cells. Any possibility their redox dynamics plays a role in these effects?
So we don't believe that the disulfide interacts with RAGE. We actually, the cell surface of RAGE has a hydrophobic patch and a electrostatic patch. And we believe so I should back up a little bit. The conformations of amidogenic species that bind RAGE are not known and could reflect several different macromolecular assemblies. There is a basic and a hydrophobic patch on the V domain of RAGE, and IAPP is basic. It has a net charge of between +2 to +4 at physiological pH, and thus it is unfavorable for it to interact with the basic patch. So we believe that it binds to the hydrophobic patch. Our tryptophan quenching experiments are certainly consistent with that. We're interested in doing mutational studies to better map the binding site. We have not done that yet.
Yeah, thank you. We have the next question is most of the questions are actually about RAGE now. Is from Wang. Great job. Just curious, where does RAGE IAPP interaction happens? IAPP has a high concentration of secretory granules. Do you think oligomerization states in cells before secretion? Do you think oligomerization starts in cells before secretion? Apologies for that.
So in type 2 diabetes, islet amyloid deposition is predominantly found in the extracellular space. And the only amino acid in the human IAPP sequence that can titrate over the pH change from the secretory granule, where it is stored with insulin, which has a pH of about 5.5, to the extracellular space where it is secreted with insulin, and that has a higher pH environment of 7.4. The only amino acid that can titrate over this pH range is a single histidine residue at position 18, which has a pKa of around 6.5, leading to a decrease in net peptide charge upon secretion into the extracellular space. So charge repulsion is expected to disfavor aggregation in the lower pH environment of the secretory granule. And aggregation is more favorable in the extracellular space where there are less repulsion interactions. Another factor that would disfavor amyloid formation in the secretory granules, interestingly, the Clark, Maranker, and Westermark labs have found that the B chain of insulin is a potent inhibitor of amyloid formation, and this may be another factor why islet amyloid formation it's disfavored in the secretory granule.
Yeah, thank you. And the next question is from Sheena Radford. Lovely results, Andy. Do you know which oligomers bind as RAGE? Is it all of them, or do some species bind better than the others? You show monomers to hexamers on the gel showing an array of species. What features of the oligomers does SRAGE recognize?
Hi, Sheena. Thank you for the question. That's a very good question, and we don't know the answer to that. Certainly, there's a heterogeneous mixture of the oligomers, and so I think it would be extremely challenging to isolate pure fractions in order to assess which individual ones may interact more or less with RAGE. We believe that hydrophobic interactions play a role in the binding of IAPP with SRAGE, and IAPP, of all the ligands, amyloid-forming proteins that have been shown to interact with RAGE, our work with IAPP is the most detailed. So we still have a lot to learn. Thank you.
The next one is from Carmelo, who says, very interesting talk, Andy. A curiosity. I really like the role of oxidation. Do you have an idea of the oxidized species, for example, proteins or lipids?
So beta cells are very susceptible to oxidative damage, and the redox state in the islets and beta cells are very tightly regulated because oxidative signaling, before it becomes a problem and turns into oxidative stress when chronically activated in the disease state, is actually a regulatory mechanism. We look at the oxidized version of IAPP, and so the peptides that we use in our in vitro studies are bioactive synthetic fully oxidized forms, and they elicit toxicity to beta cells and islets. So I'm not sure if that answers your question, but we have not looked at reduced IAPP toxicity to see if there is a difference in the oxidized versus the reduced form of the peptide.
Yeah, thank you. And this is the last question from the chat from Lothar, who asks, does SRAGE also bind oligomeric A beta in a similar manner like oligomeric IAPP?
That's a good question. So biochemical and physiological studies have shown that A beta can bind and activate RAGE in the brains of individuals with Alzheimer's disease, and that this interaction plays a role in the pathogenesis of Alzheimer's. And RAGE has also been shown to engage other proteins like serum amyloid A and prion peptides. However, it is not known what species of A beta interact with RAGE or which specific amino acids are involved in the binding of RAGE with A beta or other amyloidogenic proteins.
Yeah, thank you so much. I guess we are over with the questions from the audience, and Rams, so Sam, do you have any questions for the speakers?
Great talks, guys. Thank you very much. I have a question for Ralf. You mentioned about the peptide inhibition also occur in the membrane-mediated aggregation as well, right? Did you say that?
Inhibition, yeah, yes, they also inhibit membrane.
Do you have any insights on that? How does it work?
Actually, I think what it means is that there's a probably protofibril-like intermediate that it occurs, and once that is being generated, the peptides will bind to those and prevent the seeding from that particular species. That's our current model, and whether that actually requires the peptide to be on the membrane or not is not quite clear yet. We actually made one peptide that we think should have been closer to the membrane, but for some reason that didn't work as well counterintuitively. But so we need to really investigate this more. But the fact that they actually even inhibited the membrane-mediated aggregation was good for us to see that they go via similar pathways. So yeah, we've been mapping this before, that there really is this alpha helical form that brings everything down onto the membrane. And then there is probably the beta sheet formation occurring on the membrane, but then there's probably some dissociation of the membrane as well. So we would have to do more work to figure out exactly where it occurs. It's not as potent as all the other pathways. It's really, the other three pathways were like something stronger than what we had seen with any of the inhibitors that we had tested for the other amyloid proteins even that we were working with. But for membrane, it's just an effect that's there, but it's not like, an order of magnitude. So it's a little bit weaker.
Thank you. So Andy, it's a great talk. I really liked it very much.
Thank you, Rams.
You had the cell viability data. It's remarkable that it matches very well with your in vitro test tube kinetics. That's really remarkable, given the sensitivity of IAPP to aggregate to the cofactors and whatnot you have in the cellular conditions. So you did mention that the kinetics of aggregation is similar in the buffer and the cellular conditions. But your cellular cell viability data, each one takes some amount of time to measure, right? How do you, each data point takes some time, right? 10 hours, 20 hours? I don't know. How much time does it take?
No, it takes on the order of 3 to 5 hours. And
So that means we're not really directly correlating, is that right? To the
no, it is. So we are making toxic species in the buffer. And then at different time points, we take out the IPP aliquots and we put them on cells.
and then you measure it for after 5 hours in incubation, right?
And so the particular species taken out of the buffer, aliquoted and put on the cells, then we measure those particular species on the cells for anywhere between 3 to 5 hours. And
so you mentioned, okay, that's I got the point now. So that means it's slightly 3 to 5 hours shifted for cellular viability data compared to the THT trace. That means your kinetics is similar. That means your species would have moved on to the next stage, right?
no, so if the lag phase is anywhere between 12 to 24 hours, that 3-hour window that it is taken and put into the cell culture is almost negligible. And besides the kinetics of amyloid formation, there's also a kinetics of cell signaling, right? So, and depending on what your endpoint is, you need to incubate the cell, the peptide on the cells for a certain duration of time in order to get your readout. So for example, if you have toxic species and you want to measure oxidative stress, which is a proximate event, then you would need to incubate for anywhere between, depending on the concentration of peptide that you have, anywhere between 30 minutes to an hour. And this is an oscillating effect. So if you incubate longer, you may miss that. If you want to look at pro-inflammatory gene expression, and depending on if you want to look at the gene or the protein, you may need to incubate for 2 hours. If you want to look at metabolic dysfunction, which was what our endpoint was, and on my slide, that will require 3 to 5 hours in order to actually detect that endpoint. And apoptosis will take longer. But because the length of the lag phase is so long, we picked an endpoint to where it would be relevant to the pre-diabetes situation, which is beta cell dysfunction. And so it would take 3 to 5 hours, but it's negligible compared to the 24 hours lag phase.
I'm curious, did you try any other oligomers like metal bound or lipid bound, stuff like that?
No, we did not look at complexes with lipids or with metals. We did look at complexes with inhibitors.
Okay. And what was the result?
So we looked at,
Oh, yeah,
The-
you showed that. Yeah, yeah.
yeah,
Yeah.
this was some published results. EGCG and acid fusion are very good inhibitors, and they appear to sequester and shift the folding pathway to a non-toxic entity, whereas SRAGE is a dominant negative inhibitor of RAGE, and that sequesters it and appears to prevent it from interacting with the cell.
Great. Thank you very much. Yeah.